Well, good morning, everyone, and welcome to EAB University, a webinar series funded by the USDA Forest Service. My name is Robin Osborne, and I'm coming to you from Michigan State University, along with my EAB University colleagues, Amy Stone from The Ohio State University, and Sarah Stack and Dr. Cliff Sadoff from Purdue University. We welcome you to today's presentation by Wright State University Professor Don Cipollini, who will update us on emerald ash borer and its new target of attack, the White Fringe Tree. We previously heard from Dr. Cipollini in February of this year when he discussed the first EAB infestations found in White Fringe Tree in Ohio. As you may recall, Don is a professor of biological sciences at Wright State University in Dayton, Ohio, and is the director of its interdisciplinary environmental sciences PhD program. His research focuses on the physiology and ecology of plant defenses to herbivores and pathogens, and the physiology, ecology, and management of invasive plants and insects. Before we get started today, I wanted to remind you that your comments and questions are welcome today. Please feel free to write them in the question and answer feature, which you can find by mousing over the bottom or occasionally the top of your screen. We will make a note of all the questions, and Don will respond to these questions after the presentation so we can keep the webinar flowing smoothly. To keep these free webinars coming, we need your feedback. There will be a link provided to you in the Q&A pod at the end of the webinar for a short, voluntary, confidential survey that I hope you will take time to fill out. But if you're one of the 10 people to first fill out the survey, we will be sending you an EAB goodie bag. Either way, we hope you'll give us feedback. I will also be sending out an email after the webinar with the link to the survey in case you weren't able to access it during the webinar. For those of you wanting CEUs, if you would like a certificate indicating that you participated in today's live webinar, complete the survey and send an email message to Amy Stone at stone.91@osu.edu. Certificates will be mailed to you within a week of today's program. This webinar is being recorded and will be available for viewing soon at www.emerald-borer.info. You will also find the recordings for all previous EAB University webinars there. So I'd like to thank you for attending today. And Don, you can now share your screen with your presentation and unmute your microphone, and we'll get started.
Okay, thank you, Robin.
You're welcome. We can hear you well.
Okay, I assume that everyone can now see my screen. Is that correct, Robin?
Yes.
Okay. Well, let me take it away. Good morning, everybody. I'm glad to be back on EAB University's series to provide an update from the work that we've done on this phenomenon in the last 10 months. It's been about 10 months since I gave that first presentation. And so what I'll do today is start out, for those of you who may not have heard some of that initial information, start out with a reminder of what basically I discovered last year, and then as I go, I'll increasingly incorporate new information that we've acquired since I last spoke about this topic, and in particular to fill in gaps that were essentially left hanging the last time I presented. So we all sort of know what ash trees used to look like, right, before emerald ash borer came around. We need no reminders about this at this point, about the life cycle of the beetle and how and where it feeds, and again, what it does to most of our North American ash species. Most of you are probably also familiar with the current distribution map. This is already a few months old, showing this one overlays the current range of infestations of EAB on ash distributions, both the approximate range of natural ash trees in the landscape as well as urban tree locations that might help the beetle hopscotch across the continent. So after all of this work, that things have looked with some exceptions, looked pretty bad for ash. At least we know what to expect, or knew what to expect, until this tree caught our attention. So this tree again is White Fringe Tree, Cheonanthus virginicus. It's a native North American tree, native to the southeastern U.S., and but it's also an ornamental. So it's propagated in the horticultural trade and sold and planted in backyards and a number of the places that I'll describe today, where you want a nice, showy, small tree with these beautiful fringe-like flowers, olive-like fruits in the fall, glossy green foliage. This map shows its native distribution. You can see it's primarily a southeastern species, although it does extend over to Texas. It reaches one of its northern limits in southern Ohio, not real far away from where I am, where it is considered a threatened species by the state of Ohio. Okay, but it's in its native range. It's a fairly common understory edge type of tree, but it thrives in a variety of habitats. And of course, it's planted quite a bit far north of this range and does quite well. We have individuals in Minnesota, Wisconsin, southern Canada, and so forth that do well even that far north. So why the interest in White Fringe Tree in the first place? Well, if you look at the phylogeny of the Oleaceae, this is a summary slide indicating relationships among genera in the family Oleaceae. This is the olive family in which ash belongs. And as you see in the center, more or less, of this red circle, you see where Fraxinus, the ashes, fall out. And you can see the relationship of other genera to Fraxinus. Basically, just more or less count the number of branch points would give you an indication of how distantly related something is. The more branch points to get to another genus, the more distantly related it is. So you can see that Fraxinus is actually quite closely related to the Cheonanthus species, as well as a host of other species on the right side of this phylogeny, including Forestiera, Olea, and so forth, that I'll talk about more later. And interestingly, this slide reveals that ashes aren't that closely related to some of the other things we've cared about, like lilacs, forsythia, and privet, which have been examined for their vulnerability to EAB and so far thought to be not vulnerable. So with this in mind, this relationship in mind, and the knowledge that we didn't know much about White Fringe Tree and its use by wood-boring insects prior to last year, and I set out to see whether or not EAB was indeed utilizing White Fringe Tree. If you were to look for a host range expansion of this beetle, this would be the kind of tree you would look at. And again, I became familiar with the tree because I have one in my backyard, and I started to become aware of where there were others planted near me in my hometown and so forth, that I had a significant number to look at in the first place, and set out late last summer to do that. 30 minutes later, I come across this tree, which was along a bike trail in my hometown of Yellow Springs, Ohio, and again, showing canopy dieback, epicormic sprouting, leaf malformations, and so forth. And upon closer inspection, I first saw an exit hole right in front of me as I approached the tree. That led to bark dissections that revealed fairly recent galleries underneath that exit hole, and further dissections that revealed older galleries, and led to me being given access to cut the tree down and completely debark it, after which I found many live larvae, one of which I show in the bottom right there, that were declared to be emerald ash borer larvae. At least we were pretty certain. After this, I quickly started to write up a report and seek other data in my area on trees, again, that I either knew about or found out about very quickly thereafter. And these are just a selection of trees. In the upper left corner is another tree from Yellow Springs in a private residence, showing the canopy dieback that made me take a closer look at that tree. Found a tree over here at the Cox Arboretum in Dayton, which looked pretty good actually, but there was quite a bit of epicormic sprouting at the base. Closer inspection revealed exit holes and so forth. The bottom left tree was in a cemetery, again a popular place where these trees get planted. I didn't find an exit hole in that one at the time. I found one later, but I found this other kind of symptom that's important to note for white fringe trees, this bark swelling. It's a thin bark species. You can often see a swelling over as a response, a healing response of the tree to gallery formation. So I had cut in just a bit to see that there was an old gallery underneath that tree. So in any case, I gathered this evidence. And what really sealed the deal, however, was the fact that I revisited that original tree I found to be infested and found a dead adult beetle trapped in that tree that was certified or verified as an emerald ash borer and used to certify White Fringe Tree as a new host record in October of last year. Okay, so that basically closed the loop on that part of things. And of course, that led to this paper, which was published in January in the Journal of Economic Entomology. In that paper, I report 4 of 20 white fringe trees showing EAB diagnostic symptoms. I have to point out that revisiting those trees and now looking more closely for this old gallery formation and so forth, at least half of those trees are attacked, or have been attacked at this point. And then again, one tree that I was able to completely debark at that time produced about 20 live larvae, and the dead adult that was confirmed as EAB. So that was a big story. Most of you have heard about that. That kind of broke the internet for a while. And here's just a shot of me holding the base of that original tree that I found EAB using in some media shot in the last year. And I noticed a week ago that greenhouse grower called this find the top pest report in the past 2 years, which is an auspicious award, I guess. Nonetheless, it generated lots of attention, and of course, lots of questions emerged from that initial find. And again, we very immediately set out to try to answer as many of those questions as we could. So the first was whether or not this initial find was some sort of anomaly. Is this how widespread what was this? Is this confined to certain situations, stress related and so forth? So shortly after the confirmation of the adult beetle that I found in the tree, I got a call around Halloween of last year from Joe Boggs, an extension agent in Ohio, who said that there was a group that was working at Spring Grove Cemetery and Arboretum in Cincinnati, Ohio, who thought they found, a student group, thought they found EAB attack on a white fringe tree there. So I immediately ran down there to take a look at things, and I'll talk about what we found there, what we've found since, and beyond. So again, Spring Grove Cemetery and Arboretum is actually a very large cemetery, lots of Gothic architecture, very interesting place. The utility here is that they had 28 white fringe trees planted. We've actually learned that there are a few more than that they didn't account for originally. So it was a great site with 28 fringe trees in fairly similar growing conditions, all in the same location. Of course, there are ashes there in various states of decline as well. Another reason this site was useful is that it has a relative of white fringe tree present called Chinese fringe tree, which again is from the home range of EAB in China, and is also planted ornamentally. So 11 of these trees were planted at Spring Grove and became study subjects for us. And then extending a little bit further out in that phylogeny, there is another related species, pretty closely related to the fringe trees, called devilwood, Osmanthus americanus. It's also called tea olive or wild olive, and this is also a southeastern U.S. native that's also planted ornamentally. So 5 of these were planted at Spring Grove. And just to point that this is a far southeastern species. Its native range isn't too far out of Florida, into Georgia perhaps, but it can make it in Cincinnati, Ohio. So these things can grow far outside of their native range. Again, at Spring Grove, we did the same sort of survey that I did originally, looking for the presence of diagnostic symptoms of EAB attack, including exit holes in the upper left there. And then when I found something like that, I would just make sure there was a characteristic gallery underneath that. On all the white fringe trees, I could access Chinese fringe trees and devilwood trees. And here is again a slide just indicating that unfortunately, the large Grand State Champion white fringe tree at Spring Grove has exit holes, presumably caused by EAB in it. I also was fortunate enough to find another dead adult in a white fringe tree stem that was also confirmed as emerald ash borer. So basically tying up that loose end at Spring Grove. And the summary of that survey was that about 9 of the 28 fringe trees that we examined at the time showed EAB attack, evidence of EAB attack. None of the Chinese fringe trees showed evidence of EAB attack, and none of the devilwood trees showed evidence of EAB attack. This is in the field now, okay? And again, now that we know what to look for, and we've looked closer at those white fringe trees at Spring Grove, there are several more actually that have old galleries. So this number is probably up closer to 12 of those 28 or so. And this work has now been published as well in environmental entomology, and again, I have a student co-author on that one, Chad Rigsby. Okay, so one thing that we didn't know yet at the time was, and this was still a part of the life cycle we hadn't demonstrated yet, and that was the fact that whether live adults walk out of those exit holes, okay? We assume they do or did, but we hadn't seen that yet, okay? And so this is something certainly new that's occurred since last year. What we did after those initial surveys in the Dayton and Cincinnati areas was harvest stems from 9 of the trees that were putatively infested by emerald ash borer, and put them in rearing barrels in the lab to try to observe the emergence of live adults, just to demonstrate that part of the life cycle. So this is the tree from Cox Arboretum in Dayton. And so we more or less would discard the branches you have on the left, although we know that this size branch can be attacked, but emphasize these larger stems you see on the right. And those got placed in rearing barrels at 2 locations. This is a group that we've collaborated with, Vanessa Lopez and Annie Ray from Xavier University, who worked on both emerald ash borer and Asian longhorn beetle. This is a laboratory in Bethel, Ohio, where a lot of that work gets done. So those are white fringe tree stems in the back there, covered with a screened top to see what happens. And we got it. Okay, so those stems in that particular lab were collected and placed on February 3rd. We actually went out on Groundhog Day last year to collect them. And we had live beetles first emerge on April 8th, 2015 from those stems. There's a picture of one, a white fringe tree reared live emerald ash borer. I even had them take video of it to prove to people that they were alive. So we didn't have a great deal of emergence, but we had emergence from several of the trees, stems from several trees that we collected from Spring Grove. And again, that's been forwarded to USDA, and they've confirmed it as well. All right, now, beyond the Dayton-Cincinnati area then, very shortly after I gave this webinar last February, I received this photograph, or these photographs from, it might have actually been Robin that sent them to me originally, but then I contacted the supplier, Jim Tresaldek. This is from a white fringe tree, a street tree in Chicago, and showing diagnostic sort of EAB exit holes. And that's all I had. I haven't actually seen this tree, but that led us to expand our horizons to the Chicago area and look up places that hold white fringe trees. And that took us in late spring last year to the Morton Arboretum, where there were 16 white fringe trees planted at the time. And again, a handful of Chinese fringe trees to examine, but no devilwood. So in late May, prior to this current year's emergence date, we went and did the same sort of surveys at the Morton Arboretum that we've done at Spring Grove, looking again for exit holes primarily first, but all the other symptoms and revealing galleries underneath exit holes when we found them, and came up with another set of results that about half of those trees at Morton Arboretum were showing symptoms of emerald ash borer attack, okay? And again, like at Spring Grove, none of the Chinese fringe trees we've examined showed evidence of attack. And again, it just expanded our observation of this phenomenon a couple of states over now, and but the pattern seems to be the same. White fringe tree seems to show attack, while Chinese fringe tree does not. And again, this is part of that paper I referenced earlier with my student Chad Rigsby. All right, now since that we went to Morton, we've also continued to look at any place where any white fringe trees that we can find in the landscape or that we hear about or people report to us. And again, one goal of these sort of webinars is for any of you who have data or information or on white fringe trees, we'd love to hear from you. In any case, we've on any of these trees that we examine, we try to measure a number of variables, size variables, their growing conditions, landscape conditions, adjacency to ashes or other white fringe trees, and those sorts of things. And so using this regional inventory data, we're trying to correlate attack with some variables to see how well we can predict attack on white fringe tree based on these variables. And so this map indicates the current distribution of EAB attack on white fringe tree. The pins you see in yellow are all sites that have attacked white fringe trees. Again, presumably by emerald ash borer. We haven't collected larvae or adults from every one of these trees, but we're pretty certain what we're seeing at this point. And again, every pin here doesn't just simply represent a single tree. It represents a site that has white fringe trees, of which some proportion are attacked. I do want to point out that I had mentioned the Chicago sightings, one in the city of Chicago and one out in Morton Arboretum. But just about a month ago or so, we have new infestations reported from right under Cliff Sadov's nose at Purdue University, where my student visited, a former student of Cliff's, and finding that I think it was 3 of 5 white fringe trees on Purdue's campus were attacked. And within a day or two, we got a report from the Indianapolis Museum of Art that there were a number of white fringe trees attacked there, apparently, which we've now examined. But of course, beyond the sites I've talked about in southwestern Ohio, we've added places like Wilmington, Ohio, out to Dawes Arboretum in Newark, Ohio, and various other trees in the Dayton area that I have are not part of those other studies. So we now have about 115 total white fringe trees in this inventory. About 45 of those show symptoms of EAB attack. The bottom right here, I do indicate these pins in green are wild white fringe tree populations. They are our first wild white fringe tree populations that we've examined. They're in green because they're just ahead of the EAB wave. Believe it or not, EAB is not completely across Ohio yet in all counties equally. Although we do believe that the pin at the very most southern point, which is in the vicinity of EAB attacked ash trees, we do believe we have our first gallery on a wild white fringe tree on a tree there. Okay, so that's yet to be formally confirmed. But again, they're in green because it's not been shown that they've been attacked, but they're also mostly ahead of the EAB wave. Likewise, my student Donnie Peterson looked at some trees up in Madison on the campus of the University of Wisconsin, and they were not yet attacked, but EAB is apparently just getting to the site he examined there. So in any case, we have a number of variables measured on all of these trees. Not a great deal of predictive power came out of a number of those things, but a few things have come out to be significant so far. One is that size of the white fringe trees is a pretty good predictor of whether or not a tree will be attacked or not. So what you see here is each of these bars represents a size class. This is basal stem diameter in centimeters. And basically what you can, and then the numbers on top of each of these bars are the number of trees we have in our inventory that are in that size class. And then again on the y-axis, the proportion of trees in that size class that show evidence of attack. And essentially you can see that as you increase in size, a higher proportion of the trees show evidence of being attacked. At the very upper end, the very larger trees, you can see that it's just at about 50%, but we only have a couple trees in each of those size classes. So we assume that basically once a tree gets to be at about 10 centimeters or so in diameter at the base, a nice landscape white fringe tree, there's a 50%, if it's in a range of EAB attack, 50% of trees in that size class will show evidence of being attacked. Okay, so size, diameter is a good predictor. Another thing that's a pretty good predictor, and this is also useful in scouting, it's the same for ash trees basically, is if the white fringe trees show epicormic sprouting, we found that about 55% of the trees that show epicormic sprouting have EAB attack. Okay? If you walk up to a white fringe tree that does not show epicormic sprouting, only about a little less than 20% of those show evidence that they're being attacked. Again, this variable goes back and forth. Epicormic sprouting can both be presumably caused by attack, okay, as an indicator of attack. But in some cases, epicormic sprouting can also be an indicator of stress and damage of some sort, which can also facilitate attack, possibly. So nonetheless, epicormic sprouting is a good predictor of attack on white fringe trees by EAB, just like it is on ash trees. All right, now, now that we've examined sort of just the overall pattern of attack on trees that we know about at this point, and looked at a few predictors of that, there are still questions about how long has this attack been occurring, and is this a new phenomenon, or, for example, does attack on white fringe tree only occur after the ash resource is exhausted? These sorts of questions come up. So what we've done now to try to address some of those questions is take a look at the trees that we know have been attacked very closely and try to reconstruct their attack history, and also gauge the historical success that emerald ash borer has had on these trees. And so we went back to those 9 trees that we had harvested last year, February and March. After we were done seeing whether or not live adults came out of them, I engaged a student, Danny Thiemann, and she did the heavy lifting along with me of debarking all of these stems to look at galleries underneath the bark. And when as many of you probably know, when ash, or particularly these trees, dry, that bark gets to be very tight and it's very difficult to do. So it was a lot of work, but I think it generated lots of very interesting data. And again, the basis here is that once you debark one of these trees, if you see in this stem section I'm holding in my hand, there are several generations of usage of this tree evident right there. So I'm circling a spot right here where I had collected a live larva near the base. Here's another area where there was a live larva. Okay, so those were actively feeding in the summer of 2014. Here's a gallery that was most likely formed in 2013. Okay, fairly recent, fairly shallow, and you can see how well developed it was. Up here near the top, you see a much more well-healed over, calloused over gallery. And this happened to be the spot, by the way, where I found the adult that was trapped. And this gallery was probably produced in 2012 or perhaps earlier. We actually didn't go back to this particular tree and age every gallery, but we did do that on all of these stems that we had harvested to see if live adults emerged. And again, we can age them by looking at how shallow they were, by counting essentially the layers of secondary xylem that were covering the galleries, which is a common response to this tree as long as it's healthy enough. They heal their galleries quite well, and they get preserved in the wood. So you can age them pretty reliably. Of course, it's not perfect. But here's what we saw after doing that. So these were the 9 trees that we had harvested at the time, one from Yellow Springs, one from Springfield, one from Dayton, and then 6 trees from the Spring Grove site in Cincinnati. And this is just simply counting the number of galleries per square meter of phloem that we examined on each of these trees. And you can see there was a great deal of attack, okay? And so the Dayton tree was the most weakly attacked. It was a little bit less than 10 galleries per square meter. But we had a tree in Cincinnati here, number 3, that had over 80 galleries per square meter. Again, these are levels that rival what you might see in an ash of a similar size. So plenty of attack. A high degree of success was revealed when we looked at this as well. So what this figure shows is taking the means of all of the galleries on all of those trees. Each of these bars represents a size, width class of the galleries, okay, with the smaller galleries on top and the larger galleries on the bottom. And again, the larger the gallery, essentially the farther through development the larva has gotten. You know, there's a pretty strong correlation between gallery width and larval size. And so, again, the larger gallery, the more likely to be successful. And so you can see a great deal of these had large galleries, indicating a high degree of success of these larvae through time on these trees. And I should point out that these trees also had abundant exit holes present, which again varied, but that tree with heavy attack from Spring Grove had about 22 exit holes on it, for example. So just to give you an idea. And then this is where we really got at the history. Using our aging technique, we could see that on the bottom here, each of these bars represents the number of galleries per square meter that were produced in each year that we could examine in these trees. Again, these trees were collected in the winter of 2015, so we essentially saw what happened in 2014 and earlier. We were not confident that we could age galleries older than 2011, although we think we might have some that go back the whole way to 2010 or so. And the point of this is that you see that attack was pretty evident already in 2011. There was a great deal of attack in 2012 and 2013, and it's then declined since. I do want to point out 2 things here, that we did have this regional drought in 2012, and some folks suggested that was the reason why white fringe trees were attacked. The trees in this study were not severely impacted by that, first of all. And although drought may influence susceptibility through changes in volatile profiles and so forth, we don't think it was required in any way, partly because we have seen that the trees were attacked before 2012, and they've been attacked ever since in years of normal precipitation. All right, so what this dynamic does correlate pretty closely with are EAB densities in these areas, okay, even on ash trees. We also presume that the pattern of ash mortality in these areas parallels these results pretty well, this pattern pretty well, although we are working on that aspect at the moment. I do want to also indicate that, again, this data came from trees collected in 2015, so we looked from 2014 and earlier, but trees have continued to get attacked. I want to show a very little piece of a video that was shot last summer, July of 2015, on which I found a live larva in a white fringe tree right on camera, to my surprise. So let me just pull this up. So I'm revealing a feeding gallery that would have been produced in 2014 by an emerald ash borer larva. Oh my gosh, this is feeding this year. There's a live larva. This is this year's feeding gallery. There's a live emerald ash borer larva right there. This tree, when I did my investigation. Okay, presumably you could hear my excitement by finding that live larvae. I thought I was looking at an older gallery, but as I explored it more, I saw that that was a fresh gallery in 2015 with a live larva. And again, we presume that this is happening to some degree, or happened to some degree throughout this year, and just more or less attack on these trees we believe will parallel or correspond to EAB densities in these areas and attack on ash at the same time. Okay, so now that we've shown that attack has been occurring for a while, apparently concurrently with attack on ash, and also been occurring across across the Midwest essentially so far. The question now is, why has attack occurred? Okay, and so we've done a number of things to try to get at this question of what's causing EAB to use white fringe tree in the first place. And so again, this was again in relation to drought. Initially, folks raised the idea that drought really stressed the trees and caused them to sort of not display their normal resistance levels or susceptibility to things like EAB. And I interestingly found an image from Google Street View from 2012, the year that the drought was impacted this area. And the image, the screen capture I'm showing you here, is the original tree that I found to be attacked by EAB. When we've dissected that tree, there were already emerald ash borers in that tree. You can also see in July of 2012, sort of the peak of the drought around here, that this tree is not particularly droughted either. So that led us to suggest that drought, while it may have played some role at the time, was not necessary for attack to occur. And we've looked at other factors that attract white fringe trees, emerald ash borers to white fringe tree. And this past summer, we've done some work on volatile profiles that are given off by the bark and leaves of white fringe tree, and looked at them in relation to what susceptible ash trees give off. And so what this figure represents, this is a chromatogram that comes from gas chromatography, mass spectrometry. And the peaks on this chromatogram each represent a compound, many of them known to us, many of them unknown at this point. And this work is done in collaboration with Jim Tumlinson and Nate McCartney at Penn State University. The peaks extending upward on this figure came from white fringe tree canopies. The peaks that extend down come from a canopy of black ash. And so you can basically, by aligning these things, just simply obviously see the similarity in profiles, volatile profiles given off between white fringe tree and a susceptible ash species. You know, there are some differences, of course, but this will give us clues at what attracts EAB to white fringe tree, as well as what seems to be the important shared things that might attract adults to ash trees as well, that might be useful in trapping. In red, by the way, I point out, one clear thing, methyl salicylate, production of this compound methyl salicylate, that both black ash and white fringe tree share. And when you compare white fringe tree to a resistant ash species, Manchurian ash, which is not very attractive to adult EAB, even though it's one of their normal hosts, you can see that that's something that's basically missing in Manchurian ash, but present in White Fringe Tree. And if you look at the far right of this particular chromatogram, there's also this a couple of metabolites here that are very highly produced by Manchurian ash and not produced by White Fringe Tree. And that actually may be a deterrent that Manchurian ash produces as a result of its coevolutionary history with emerald ash borer, to have it avoid being attacked in its home range. So this is still fairly preliminary data, but we've now, and again, I should point out that this is data taken from healthy trees. And so right now, we believe that the main reason an emerald ash borer uses White Fringe Tree is that they smell just like an ash tree that they like to eat. Okay, now backing up a second back to, but also relating in host quality and chemistry a bit. Our field data from the Dayton area and Spring Grove and Morton Arboretum and so forth suggested, if you recall, that White Fringe Tree gets attacked pretty regularly, while species like Chinese Fringe Tree shows no apparent evidence of attack in the field. And again, Chinese Fringe Tree is a close relative of White Fringe Tree. When you get a result like that, you don't know, however, whether or not the adults had placed their eggs on Chinese Fringe Tree and the larvae failed on the tree, or whether or not they're not even placing their eggs on the tree, or what. So we looked at a piece of that, and again, remember, devilwood was also something we looked at Spring Grove Cemetery. We looked at basically how well emerald ash borer can perform on alternate hosts when you place the eggs directly on the trees, to at least get at that part. If an adult were to lay an egg on these trees, can the larvae survive? And so we set up stem bioassays. These are cut stem bioassays done in a laboratory. This happens to be a stem of Chinese Fringe Tree. We get eggs of emerald ash borer sent to us from the Brighton, Michigan APHIS lab. And again, these are emerald ash borers that have been raised in captivity on ash, okay, their entire life, all right? So they're essentially fed ash trees. And you can place whatever stems of whatever species you're interested in the incubator. These things are set into moist floral foam, and then you artificially inoculate them. That's a layer of parafilm covering the egg just to hold it against the bark until it can hatch and enter the bark. So what did we find? In the upper left corner, and again, this is something I had left hanging at the end of my webinar last year. We have green ash in the bioassay as essentially a positive control. Of course, after 40 days, this is a 40-day bioassay, larvae performed very well, reached the 4th instar, nice and healthy. Basically, every egg that we placed on those green ash stems yielded a 4th instar larva. On White Fringe Tree stems, essentially the same was true. Every egg we placed, nearly every egg we placed on White Fringe Tree, turned into either a 3rd or 4th instar larvae. They were smaller. They were about a 1/3 the size at 40 days, but they were healthy and they had more time to go and would have reached, presumably would have reached the prepupal stage, maybe a little bit behind green ash in this bioassay. On Chinese Fringe Tree, however, there was no development. So what I'm circling here is a gallery, not a larva now, just a gallery that the larva excavated but died. Okay, so that's about the longest galleries we saw in this bioassay on Chinese Fringe Tree were only about 2 centimeters long. And there was no survival, and we had no larvae to recover from these trees. So they just simply, they tried, but they couldn't make it on Chinese Fringe Tree. Devilwood, however, while only a few larvae made it, a few larvae made it on devilwood. Okay, and so these were small. They were probably 2nd instar size, but they excavated galleries up to about 50 centimeters long. You can see here. This is a pretty thin barked species. It's a lot more thinly barked than, say, White Fringe Tree. And that was intriguing. I still would not say that devilwood is vulnerable given this level of success, but this is only the second species outside of white fringe tree now that we've seen larvae live this long outside of the ash genus. All right, so there you have it. Devilwood actually produced a couple of larvae that survived a 40-day bioassay. So anyhow, this is just a summary. I don't really need to go through this, but that larvae did very well on green ash, did almost as well, although smaller, on white fringe tree, could not survive on Chinese fringe tree, which again corroborates our field evidence that Chinese fringe tree doesn't show evidence of attack in the field. And again, we had a couple. We had 3 larvae make it on devilwood, but they were quite small. Now we've done a follow-up bioassay. Again, this paper, by the way, or this result is published in Cipollini and Rigsby, Environmental End. We followed up on that data with another short-term bioassay where we paired these two. This nice study we now have of a resistant and susceptible and closely related ash species and a resistant and susceptible closely related fringe tree species. This was a very short-term bioassay with thin stems, and what it revealed again was that nothing could make it on Chinese. A decent proportion survived on white fringe tree. These were smaller stems, they didn't last as long. Larvae didn't do real well on Manchurian ash, which we know, but there was some survival and growth. But success on white fringe tree was similar to success on black ash in this particular short-term bioassay. And importantly, success on white fringe tree was better than success on the native host of emerald ash borer, Manchurian ash. So this is just to put things in perspective now with more ash species involved. But again, Chinese remains basically completely resistant as far as we can tell. Why is this now? Okay, well, we've started now to profile the putatively defensive chemistry in the phloem of fringe trees. Again, we've been doing this with collaborators Enrico Bonello and Dan Herms and so forth for years in ash species, trying to find differences between resistant and susceptible species. If you look at the bottom of this, these are chromatograms that come from liquid chromatography. Again, very similar. Each peak represents a metabolite. At this point, we haven't picked through and identified everything just yet. If you look at the profiles of Manchurian and black ash at the bottom, however, you see how similar they are in their phloem chemistry, at least this kind of chemistry, making you see why it's been difficult to pick out why Manchurian's resistant and why black ash is susceptible. But Chinese and white fringe tree show a couple of interesting things. Again, they show similarities, as they should. But there are 2 metabolites I've highlighted. Number 1 here is a metabolite called oleuropein, common more or less to the Olaceae. And number 2 is pinoresinol, okay? And you can see the difference in ratios of those 2 peaks in white fringe tree to what you find in Chinese fringe tree, because otherwise the profiles look pretty similar. This may point to something interesting, okay? Again, this is all very preliminary. And you can also see that while the ashes have oleuropein in these particular profiles, they have very little of this pinoresinol. All right, so this is brand new data. This is one place we'll be moving forward with in the near future. Okay, so that's a bit about adding in some new chemical data. But again, all the while that we're exploring how well and extensively the EAB can use white fringe tree, we're also interested in other relatives that it may encounter. So we're both looking at new potential hosts and maybe also revisiting some things that have been examined in the past. We're still not worried about lilacs. In fact, I've placed eggs again on lilacs this summer, and you get the same result as we saw with Chinese fringe tree. Simply, they try to eat it for a day or two and then they die. So we're still not worried about lilacs. Again, they are distant relatives of ashes in the fringe trees. However, there are things on the right side of this phylogeny, again, to be concerned about. And one close relative that's shown right beside the fringe trees here is Forestiera, which is also a small tree, a shrub, native to the southeastern U.S. I guess before I get to Forestiera, however, there is one other species of fringe tree in North America. Okay, it's called pygmy fringe tree, and it's a very close relative of white fringe tree. It's restricted to the Sandhills region of central Florida. And here's a picture I've taken of one. This is actually a small specimen of pygmy fringe tree. They do get a lot bigger than this. It more or less looks like a small version of white fringe tree. And again, it does get up to the sizes that we know can be attacked in white fringe tree. Here's a 5-centimeter diameter pygmy fringe tree stem, which is well within the range that we've seen attack on white fringe trees. To that end, here's a branch, this might be surprise some people, with a fully mature feeding gallery from EAB and an exit hole that's only 1 centimeter in diameter. Okay, this came off of a white fringe tree in Springfield. All right, so these trees are in the size of being attackable, and they're in the vicinity. Ashes do extend down into this part of Florida, and white fringe trees extend into this part of Florida as well, so they will eventually encounter EAB. We were able to acquire some stems of pygmy fringe tree from collaborators in Florida at Archibald Biological Station and do a couple of short-term bioassays. Again, these are thin-stemmed. When you do cut-stem bioassays, you can't run them for very long, especially with small-stemmed individuals. But this was mean gallery size, gallery length on white and pygmy fringe tree at 2 weeks and 3 weeks in a short-term bioassay we did this past summer. And essentially you can see that they can produce galleries of very similar sizes on pygmy fringe tree as they do on white fringe tree over this time period. So we are at this point assuming that when EAB does come into contact with pygmy fringe tree, that it can be attacked, and they probably will be able to use it with success. It's a federally endangered tree, unfortunately, so it's perhaps even more endangered now. Okay, the other species then of interest are the Forestieras, commonly called swamp privets, although they are not very closely related to the ornamental privets, European and Asian privets that are in the trade and are sort of invasive species. Up in the upper left, you see the distribution of this particular species called eastern swamp privet. It's kind of a wetland, small shrub or tree. You can see from this picture the similarity it has just in morphology with white fringe tree. And so we'll be looking at that one quite soon. Within a couple of weeks, we'll have data on that. And we are also looking at cultivated olive. This is something I mentioned last year. And we've started work on this, but at this point, I have nothing I can report. But the next time I perhaps have an opportunity to do this, we'll have data that we can share on some of these new species in terms of the ability of emerald ash borer to utilize them. All right, so I covered a lot of ground there, but just to give you kind of an overall summary, we have about 115 trees in our ornamental fringe tree inventory. We have others in the wild inventory that aren't included in this right now. And about 44 of those trees show evidence of EAB attack. We found, larvae and adults in some of those, but most of these are based on diagnostic symptoms of exit holes and galleries. Attacks now been found across the Midwest, Ohio, Indiana, and Illinois, really in any place where white fringe tree has encountered, or EAB has encountered white fringe tree at significant densities. And we're also finding that white fringe trees appear to get attacked in a given area basically as soon as ash trees get attacked. It is not the case that they move to white fringe tree only after ash trees have been devastated. Even in my hometown, there are healthy ash trees that remain, and it's not the case of them only moving to white fringe tree after they've exhausted the ash resource. Although stress may play a role in the attractiveness of beetles to these trees, we think they use actually shared volatile cues with their normal ash hosts to find the trees, and the larvae can apparently tolerate the chemistry of white fringe tree fairly well, perhaps not as well as on our most susceptible ash trees, but they do perform better on white fringe tree than on their normal resistant ash hosts from Asia, like Manchurian ash. Chinese fringe tree is, however, apparently completely resistant, and it will be useful to really pick through to find out what makes it resistant and white fringe tree susceptible, given their close relatedness. And again, we have a very nice parallel now with Manchurian and black ash and Chinese and white fringe tree that we will be continuing to run with in the next couple of years. And then finally, we are also continuing to explore both the hosts we've talked about here and new hosts based primarily on that phylogeny indicating the relatedness of species. And again, we're still not very worried about the big popular ornamentals that have been examined in the past based on their relationship with the fringe trees and ashes. But we will continue to look at whatever we can find. Okay, so I want to point out before I put up a slide of acknowledgments that, again, we're interested in any information at all about white fringe trees or these other hosts. If you know of them, where some can be located, if you see anything, we'd love to know about it and get it on our inventory and take some measurements and try to really continue to make sense of this phenomenon. All right, and so with that, this is just my simple acknowledgement slide. Lots of the people, we've had lots of help. People have been very helpful at sites and with information and expertise. And again, I want to acknowledge funding from USDA APHIS that has helped us really run with this work in the past and current year. So I want to thank you all again for listening, and I guess I will pause and be ready to take questions.
Thank you, Don. This has been quite an update. I mean, there's a lot of different things that have happened, so I'm glad you were able to join us and do this today. We do have a question. Is the predation of white fringe tree by EAB a response to declining healthy ash population, or will it feed equally on other species? I think you kind of answered that. This was a little earlier on in the presentation, but I didn't know if you maybe if you had anything to add to what you said.
Yeah, sure. I did address this a bit, but I want to reiterate that what we believe is that as soon as emerald ash borer shows up in an area, primarily on ash, which are much more abundant than any white fringe trees that might be around, if there are white fringe trees in the vicinity, they will find them at about the same time they find ash trees. Again, we believe they're using the same signals to find white fringe trees that they use to find ash. And those signals seem to overlap significantly. So it is not the case that they move to white fringe tree after running out of ash. And in fact, we have plenty of evidence from our historical reconstruction that they were starting to attack white fringe tree in these areas the same time, essentially, they were starting to attack ash trees.
Okay, that sounds good. Cliff has posted the survey for today on the Q&A pod. I just wanted to let you know, and also I have posted it on the webinar chat feature. If you see that, you can get into that in the bottom of your page for all you participants. And B. Miller thanks you for your answer to the question. Are there other questions that anyone would like to have answered right now? You did get Don's contact information, So if you wanted to contact him via email, I'm going to put that here in the chat pod as well. So, and
Robin, I have one point I could talk about that I didn't address. That
okay.
a question that's has come up throughout this phenomenon, I guess, would be the role of grafting. And I might have addressed this last year, but the early ideas were that the reason white fringe trees might be getting attacked is that these ornamental trees are all grafted on ash rootstocks. And if you look at very old horticultural literature, even going back to the 1800s, there are some suggestions that white fringe tree could be propagated on ash rootstocks. And if you search those terms on Google, that's what you get. But if you look at the more modern literature and on horticultural practices and propagation of white fringe tree, it's almost all seed production, seed propagation. They don't root from cuttings very well. They're very difficult to root. And I can also say that I cannot see any evidence, and believe me, we've looked at every tree, of any of the trees that we've examined being grafted on ash rootstocks. So we have absolutely no evidence that any of these trees that we've examined were grafted. And on top of that, grafting is not at all a common practice for propagating white fringe tree, grafting on ash rootstocks at the very least. So I just wanted to make sure I mentioned that, because that still comes up every now and then.
Okay, sounds good. We're still having a little bit of an issue here with the survey slide, but I just want to let everybody know that you will be sent that with the email that I will be sending out after this too. So don't think you're not going to get it. I also wanted to say that I know some people are saying that they were not able to see that video. So Don, I'm going to get that link and I'm going to provide that to our participants in that email so folks can look at that. And I think I can add it to the recorded webinar too with a little editing, so we can do that.
Okay, yeah, the slide, I have the links on, let's see, it's slide number, where is that? Slide number 41 has the links. There are two links to the same video, essentially. One's on YouTube, so it's easy enough to go to YouTube and find it.
Okay, that sounds good. All right, now that longer link on the chat pod, folks, that one does go to the survey, and if you want to look at that, it does go to it right now. Let's see, do we have, okay, anyone is, Wynn Dunwell asks, is anyone grafting white on Chinese fringe tree? I don't know if you saw that question.
Again, I know of no evidence of that at all. Okay. And I'm not sure why one would do it, even, necessarily. So, okay. Yeah.
All right. If there's any other questions, I suggest that you get a hold of Don, or you can answer the email that I will be sending out, and I can get other information to him. Is there anyone else who's got questions here? Let's see, we've got, I've been told that the state of Indiana blocks the YouTube, so I'd have to have a special status to get it from YouTube. So if I can embed this somehow, we can figure out what to do here. So we'll try to figure out something.
Yeah, I can help with that, Robin, if necessary.
Okay, sounds good. All right, folks, I want to say thank you all for joining us today. And like I say, this recorded webinar will be available for viewing later today. And thank you so much, Don, for updating this. This is very interesting on as far as what's been going on. Kind of crazy.
Certainly,
especially all the different, all the different species that may be attacked by EAB.
It does reopen the question of host range for this beetle, certainly.
Yeah. All righty. All right, folks, I'm going to be closing down the meeting here in just a couple seconds. Thanks again to Don and everyone else for participating, and I hope to see you all again on the next EAB University presentation. Thanks again, Don.
Thank you.